everyone. Today we're going to do one of my favorite labs, which is about animal reproduction and development. This is an important topic for before we start diving into all the different phyla among the animals, because the characteristics that are defining in terms of evolutionary patterns that we use to divide the phyla up into major groupings are almost all due to characteristics that they share based on their very early developmental patterns. So when we're talking about development, that is a process that is going to happen in between the zygote and the multi-celled adult. So when we think about what has to happen in between the single-celled zygote and the multi-celled adult, not only does it have to grow, which would be an increase in size, but it has to develop. And by development, we mean that it's going to have new body structures form, and it's going to have changes in the function as well as the structure of those characteristics. So first we want to have a really good definition of development. And so that's going to be a change in structure and or function over time. And specifically in the context of this lab, we're talking about development happening between egg or zygote and adult. We use the word development in things like economics or in politics when we say there's a developed country. And that word also refers to the process that's happening that's changing the structures and functions of that country. But specifically, we're talking here between zygote and adult. All right, so that's our definition of development. So animals are multi-celled. That means that some multiplication of the cells is going to have to happen in order for them to grow into an adult, but also that the major body structures are going to be built by this process. So we talked in lecture already about the idea of sexual reproduction, that the male and the female have to get together. There's different ways of doing this, and that we can have external fertilization versus internal fertilization. Here's some frogs. The frog has laid the eggs outside of her body, so the male can put the sperm near them, so that's external fertilization. And these frog eggs are going to start, if they get fertilized, develop in the pond. So we have a little baby frog is going to be called a tadpole. It looks different than the adult does. So when we look at that kind of life cycle where the juvenile starts out looking very different from the adult, and then its body goes through some process of extra change, a lot more change than other types of organisms do, we say that there's a metamorphosis happening, so that early developmental stage, the juvenile doesn't look like the adult. The metamorphosis is the process that happens to convert that individual from looking like the juvenile stage to looking like the adult stage. And in frogs, that juvenile is going to be in the pond, whereas the adults are going to live outside the pond in the upland. So that larval stage, because the individual is small, may need to have a different habitat than the adult does in order to maximize its ability to survive through that challenging period when it's small and relatively defenseless. All right, so when we look at frog as an example, as it starts out, it looks like a tadpole. If you don't know that word, this guy, we call it a tadpole. I don't know why, it's a funny word. And it's going to go through metamorphosis. All right, we also have a butterfly is going to start out as a caterpillar. And it's going to go through metamorphosis to become an adult. But when we look at other kinds of animals like this chick, it basically looks like a chicken when it starts. And so it's just going to increase in size. Now it's doing development too, but it isn't going through this complete transformation. So when we look at a baby, the baby, can we tell it's the same species as the adult? If we can, then we say it has direct development. And that's another way of saying it doesn't go through metamorphosis. It goes directly from juvenile to adult. If we have direct development, then the opposite of that is indirect development. So both of these organisms up here have indirect. You start out one way and you take a detour. And then in general, what we're going to call these life stages, the juvenile, when it's going to go through metamorphosis, so a juvenile that doesn't look like the adult is going to be called a larva. Right, so we could say both frog and butterfly have a larva that starts life out and goes through metamorphosis to become the adult. Whereas the chick is just a juvenile. It's just a small version of the adult. Right, so those are major categories of types of development. Although very, very early in development, all these organisms are going to have processes that look pretty similar. So we're going to look at that very, very early development today. We're going to look at a couple of different species and look at how development happens to see some differences across the animal world. And we're going to look at chick and frog because those are vertebrate animals that are very similar to humans. So we know a lot about how human development happens because we've studied these model organisms or ones that we use as an example that's similar to human development that we can learn a lot from. Right? So we have frog, we have chick, we also use mouse and rat. Other organisms that are used frequently to study development include sand dollar or starfish, and so we'll look at those today. And also a nematode worm called C. elegans. We'll see the example of that today as well. And also fruit flies, which is what Dr. Miller uses to study development, but we don't have any examples in lecture. All right, so when we look at sand dollar or sea urchin, those are convenient to look at because like many marine invertebrates, they're gonna release their eggs and sperm into the water, and that makes it really easy for us to take them into some kind of container, put them on the microscope, and look at them. And so if we were in class, we would have live sand dollars in lab. I would be injecting them with a potassium solution, and that would make them release their gametes. So when we have egg and we have sperm, another way to talk about them both together is called them gametes. So we would have a little beaker with eggs in it. We would have a little beaker with the sperm in it. The eggs are so big in sand dollar, which is another nice reason to study them, that they'll look like a little pile of sand grains on the bottom of our dish. So you can see them with the naked eye. And put them under the microscope, we're going to be able to see the egg cell here. And then if we mix sperm with them, these little tiny kind of toothpick shaped structures, those are the sperm, and we would see them, they would be wriggling because they can swim with their flagella. And then the egg cell is surrounded with a gelatinous coating, the jelly coat, and this particular species puts these purple dots of pigment in that jelly coat. So we don't know why, but it makes it really easy to find these. You can see these egg cells using the 4X objective lens on the microscope. So if we put egg and sperm together, within 5 minutes, if the eggs are viable and the sperm are viable, then we're going to see this fertilization envelope form. So when we talked about this in lecture, we're going to have this fertilization envelope. Right, so we see this clear window almost around the egg. So that's created when the fertilization happens, and that fertilization envelope lifts off from the surface, and it creates this space around the egg cell membrane once the zygote is formed, and that prevents a second or more sperm. Right? So it's a blocked polyspermy. It's a permanent one. We can see this. It looks like we almost cracked an egg into a pan, and we don't have the burner on yet, so we see the yolk, and then we clearly see that We can see that clear white of the egg around it. Even though it's clear, we can see the edge of it. And this happens, I've been fortunate enough to actually see it happening a few times out of all the years I've been teaching this lab. And you can see where the sperm actually fertilized the egg, because the fertilization envelope will pop away from the surface of the egg in one spot, and then it will spread as it's going around the egg. So within 5 minutes or so, we'll be able to see visually that we have fertilized eggs on our dish. So rapidly, what starts to happen after fertilization is cell division. So we have this rapid cell division that happens right after fertilization. And it happens so fast that the cells don't have time to grow in between cell division. So we start with this immense egg cell. Remember, it's big enough to see with the naked eye. And for a while, it's going to start dividing and then dividing and then dividing and dividing so rapidly that the cells that are the subsequent daughter cells are going to get smaller and smaller and smaller. Right? You can't do that forever, so it'll stop doing that at some point. But this early series of divisions where the cells are getting smaller, so this whole series of divisions that are shown on this slide are called cleavage. So cleavage is the first series of cell divisions that occur after fertilization. And the cells, the daughter cells, get smaller and smaller. We can see the fertilization envelope is still around each of these embryos. So we can see as we go from one slide to the next that those cells are getting smaller and smaller. One cell turns into 2, 2 cells turns into 4, 4 cells turns into 8. So the cell has divided 1, 2, 3 times to give us 8 cells. And we can't see all 8 cells here, but we can see 1, 2, 3, 4. And these are smaller than the cells that we see in the 8 cell stage there to the left. And so behind them, right, so immediately behind them, we would see cells 5, 6, 7, and 8, but they're sitting on top of one another like the bricks on a Rubik's Cube, so we can't see them in this picture. If this embryo were turned on the side, we would be able to see it, but we're looking at it from the top. Right, once it gets to the 16 cell stage and beyond, it's harder to count up the cells. It looks more like a ball of cells rather than specific individual ones that we can see that are arranged in this geometric pattern. Right, so this sand dollar embryo is going to continue growing until it becomes a larva. So sand dollars are going to have a metamorphosis, and that means that the embryo is going to develop into a weird looking thing that doesn't look anything like an adult sand dollar, and we're going to call it a larva. So all the echinoderms, the phylum of animals that sand dollars are in, have sort of crazy looking larvae, and sand dollars larva is called a pluteus. And for a while it's going to grow, and it's going to have four little stiff arms sticking out. Four. And then it's going to grow some more and end up with 8 arms sticking out. But notice that this larva is bilaterally symmetrical. It has a little bit of an uneven shape. Now it's going to be swimming around in the water. So it's going to be in the plankton, and it's going to be able to, it's very tiny, it's going to be able to feed on single-celled algae, phytoplankton. And then when it's large enough, it's going to settle down to the bottom. Rather than swimming, it'll let itself sink. And then it'll go through metamorphosis and turn into that round, flat sand dollar. So in between the time that it is a zygote and it is a larva, we're going to look a little more detail at the things that happen in that process. So we already looked at early cleavage, where we have a zygote. It goes through a series of cell divisions. We show the 8 cell stage here. It's going to keep doing cleavage. And then we have a stage that is in between the 8 cell and the blastula. It's called a morula. And the morula is where we have enough cells that it forms this solid ball. So it's like all the cells are just packed together. So that's a solid ball shape. And as cleavage continues, the cells are going to get small enough, and they're also going to organize themselves so that they're around the edge of a circle and there's a hollow space in the middle, right? And that hollow ball of cells is going to be called a blastula. That hollow space in the middle is going to be called the blastocele. So that word seal or coelom, we had a spongocele in our sponge animals from last week. That's that hollow space that's inside the middle of the sponge. So that just means it's an empty space. So if we slice through our blastula, it's like slicing through a soccer ball, that there's an empty space in the middle and the cells are forming around the outside. All right, so we say that each of these stages or the structures that we can see and describe, we're going to say that those are stages of development. So once we form a zygote and it starts to divide, then we can talk about the thing that is happening, or we can talk about the actual individual. Right? So the individual is going to be called an embryo. And as it continues through this process of development, we can say this is an embryo that's at the 8-cell stage of development. This is an embryo that is at the blastula stage of development, right? So after it becomes a blastula, the next step is a process called turning into a gastrula. All right, so all the cells in the blastula are the same as one another. So differentiation hasn't started yet. Differentiation is where cells turn into different kinds. Right? They become different from one another. And what's happening during gastrulation is that these cells are starting to form this dent or dimple on the side, one side of the ball, and they start to push in. Right? We talked about this already when we talked about cnidarians, that this embryo is going to start to have this inside versus the outside. So the inside, this space that forms in the inside of this sac-like shape is going to become the gut of the embryo. Right, and so if this is a cnidaria, we put some tentacles on the bottom of it, we'll have a jellyfish, or if it's upside down, we would put tentacles on it and it would be a polyp animal. Notice that we still have a blastocele. It's been pushed out to the edges, but it's still there. This opening, this little round circle that we'd see if we looked at the end of this embryo, is a hole or a pore. It's like the mouth, right? So our mouth is our opening at the end of our digestive tract. That's called the blastopore. So this tube, or this space that is going to become the gut, right? So that becomes the gut, is going to have a name as part of the embryo that's called the archenteron. And the cells that are lining the archenteron are going to be this different, separate kind of cells that have different functions. So they're going to start being able to secrete digestive enzymes, and they are forming a layer of cells that is called the endoderm. All right, endo meaning inside. The ectoderm is the layer of cells on the outside. Right. Now, if development stops here, like it does for cnidaria, we're going to have what's called a diploblastic organism, because it only has these 2 tissue layers. And it doesn't develop that third one, it stops here. Now, if gastrulation stops here as well, we're going to end up with an organism with an incomplete gut. And in our example of an organism that has development that looks like this, and then it stops, continuing on these processes, and just starts to grow into an adult, is going to be cnidaria. So cnidaria is our phylum that is diploblastic with an incomplete gut. All right, so that's a little review. Now, when we look at our sand dollar, it actually is going to have the same stages: zygote, it's going to go to 8-cell, it's going to go to blastula, And we have an early gastrula where that dent is starting to form. The later gastrula, though, we're going to see that not only does the gut continue to form until it becomes a continuous tube that goes all the way through the body, but we're also going to find these cells that start to fill up the space in between the endoderm and ectoderm. And so as gastrulation continues, this tube is going to keep extending until it joins with the other side, and we'll end up with another opening on that end. So this side down here is where the blastopore was. We have the archenteron is developing inside of that gut. And then we have this third tissue layer that's going to fill up some of the space in between the ectoderm and the endoderm. And that's going to be mesoderm. So that's the third tissue, right? So this organism, it's triploblastic. It has 3 tissue layers. It has a complete gut. And we're going to define a complete gut as having 2 ends that we're going to call the mouth and the anus, and food is going to go in one end and get digested and processed, and then waste is going to come out the other end with one-way flow, right? That's what we have. All right, so when we look at these tissue layers that form very early in development, they go on to form particular body structures in all animals. So endoderm is going to go on to form the digestive tract and the structures that are around it. The ectoderm is going to form the outside of the body. In vertebrates, it also forms the nervous system, and we'll see how that starts to happen in a little bit. The mesoderm is going to form the tissues that fill up the middle of the body, muscle, skeleton, and connective tissue. And every embryo that starts out going through development, as soon as it's fertilized, we can tell which cells in that embryo are actually going to go on to become particular structures in the body of an animal, right? Plant development doesn't work like this, but in animal bodies, every cell, once development starts, is going to divide and grow and turn into, eventually, different structures in the body that is utterly consistent from one individual to another, as long as development is proceeding normally. And so we say that each cell in development has a fate, right? That it's destined, that it's going to turn into a particular structure. And this research was first done on a worm, a nematode worm. We'll come to them next in a couple labs, nematode. So that's the phylum. They're round worms. And it's called Cenorhabditis elegans. Cenorhabditis is a mess to spell, so I'm just going to say C. elegans. So often we'll talk about C. elegans. It's another model organism for looking at development. So these worms are microscopic, or they're barely visible to the naked eye, and they have about 1,000 cells. Right, so it's much easier to draw a map, a fate map of once development starts as a zygote and the cells divide into 2. You can see from this diagram, what do those cells turn into? So we can see that a whole bunch of those cells end up turning into the intestine. And then as the cell divides and divides, it turns into different parts of the body. And we can see where these cells end up in the adult structures. All right, so the important concept here is not, the specific details of this for C. elegans, but the idea that in animals, the developmental fate of every cell is set once development starts, and that if something goes wrong at any step in development, all of the subsequent structures that would be built by those cells are going to have some abnormality in them. All right. So when we look at those 3 different tissue layers, there's a couple ways that they end up being arranged in the body of the animal. So we want to look at this, and we're going to be looking at, is there a space inside the body, or does that mesoderm completely fill it up? And if there is a space, a coelom, how is it with respect to the muscle tissue that's there that is formed from the mesoderm? So we have 3 different phyla that are examples. So we looked at Platyhelminthes last lab. Right, the flatworms. They are triploblastic, right? They have an incomplete gut though. And so we're only going to be talking about these body spaces or coeloms once we get to triploblastic animals. So these are only for triploblastic. All right, so as the mesoderm is forming, it's shown in red on this slide. So the mesoderm is going to be filling up the middle here. My red pen isn't showing. We have this yellow circle. Now the yellow circle is representing the gut. So the gut is going to be, even though it's an incomplete digestive tract, at some point in the body, we're going to slice through and we're going to see this space. So if we're just drawing a cartoon of what this body looks like, we're going to have the gut right in there. And the mesoderm is going to fill up all the space between the endoderm and the ectoderm. So the body's solid, right? So we call this acoelomate. It's a body that does not have. So the prefix a in front of a word means it doesn't have it, or not, or none, or no. So acoelomate means it doesn't have any body space, right? Now, when we do have animals that have a body space, it turns out there's 2 ways it can look. If we, again, draw this cartoon of what this looks like. Now, our next phylum is the nematodes, Nematoda, like C. elegans, roundworms, that they are going to have the gut, right? And it's going to have a space inside of it, the lumen of the gut. That's where the food goes. And then there's going to be a space. Their body space is going to be between the gut and the mesoderm. So they do have a body space. But when we look for where the mesoderm is, it isn't filling up the entire body, but it's just next to the outer body wall. It's just next to the ectoderm. Right, remember that the digestive tract is endoderm. So another way of thinking about this kind of body plan, these organisms are called pseudocoelomate. It's a sort of a coelom, and that is because it's only lined on one side by mesoderm. So the mesoderm is basically stuck to the inside of the ectoderm, and it's not touching the endoderm. If we look at an earthworm, this is in the phylum Annelida. Right, it's going to have a true coelom. So sometimes this is not just coelomate, but eucoelomate. It has a true coelom, and that is going to be, we have the ectoderm, and we have the gut, and we're going to have a little layer of mesoderm around the gut, and we're also going to have a layer of mesoderm inside the ectoderm. All right, so you can see that the space, the coelom, is lined on both sides by mesoderm, whereas in pseudocoelomate, there's only mesoderm on one side of it. All right, so these are just things that you need to remember, that we have different phyla, and they can be eucoelomate, pseudocoelomate, or acoelomate organisms. All right, another characteristic that is going to be different in major groups of organisms is whether they're a protostome or a deuterostome. And this has to do with what happens to the blastophores. So we have organisms, once we have triploblastic organisms that have a complete gut, we're going to be able to divide them into these protostomes and deuterostomes. Right, so this is an early gastrula. And Remember, this guy here, this ring or space, is called the blastopore. Protostomes, once that gut continues, oops, it's kind of messy. All right, so the blastopore is still down here. And this tube is going to go all the way through the body, so we have a complete gut. And the fate of the blastopore in protostomes is that this is going to become the mouth end of the animal, and this end is going to become the anus. So that's not too surprising. The part that's formed first is going to be the head end of the embryo. But it turns out in another major group of organisms, the deuterostomes, so proto means first, mouth first, deuterostomes means mouth second. So we're going to start with the embryo in the same orientation. The gut's going to grow all the way through it. This blastopore, though, is instead of being the mouth, that's going to turn into not the head end of the animal, but the other end of the animal. Right, so what we have, the difference between the protostomes and the deuterostomes, is that the orientation of the body of the animal from early development is 180 degrees opposite. Right? So this is an incredibly trivial seeming, but it turns out it's a very important and meaningful characteristic in development. So once we had defined a lot of the differences between phyla based on development, because microscopy was the major tool of studying development for a long time, and we divided organisms into different kinds, in part based on their mode of development, then we went on and found DNA sequence data, and we looked at how closely groups were arranged with one another based on their DNA sequence data. And most of the characteristics that have to do with development turned out to be consistent with the groups that we formed based on DNA sequence data. So the main idea with that is that what happens in development is a very powerful predictor of how closely related different taxa are. The way their bodies are formed is patterned by their DNA. They have shared DNA, their bodies are going to be patterned in the same way. So protostomes and deuterostomes not only have the mouth versus the anus forms differently, they have differences in cleavage timing and cleavage pattern. Right. So remember that each cell has a fate in development. It turns out that deuterostomes set the fate a little later. Right, and protostomes, we say they have determinate cleavage. Now, deuterostomes, the fate is determined as well. It just happens a little later. So the fate is set earlier. Right, what that means is if we take an 8-cell embryo of a protostome and we separate it out into the 8 separate cells, it'll die. But if we take an 8-cell embryo of a deuterostome and we separate the 8 cells apart from one another, each one can still turn into a whole complete embryo. So we'll have a set of 8 identical octuplet sand dollars, for example. Those cells won't die. Their fate hasn't been set yet. They can reset to the beginning of development and start over again. So it's not much longer. It's a few cell divisions later. Right, and then the last characteristic that we're going to talk about is how the cleavage pattern progresses. In radial cleavage, we looked at sand dollar and we saw that when it goes from 4 to 8 cells, that those cells are stacked up right on top of one another. Right, it's as if we had if we put two squares and they're just sitting right on top of one another. That's called radial cleavage. Protostomes, the first set of cells that divide, and then the second set of cells that divide, are a little offset, or they're twisted from one another. Like if you take a Rubik's Cube and you turn it, right? So they're gonna start to form a spiraled shape. And so we say they have spiral cleavage instead of radial cleavage, right? So when you look at the 4 and 8 cell embryos of protostomes versus deuterostomes, you can see in the embryo that these differences happen, and we'll do that in lab today. So our sand dollar and sea urchin or starfish is an example of a deuterostome. And we have a ribbon worm, which is called Cerebratulus, which is an example of a protostome that we're going to look at as our example of spiral cleavage later on in the lab. All right, so one of the reasons why we study sand dollar and sea urchin is not only that it's easy to get their sperm and eggs and to watch them go through development, but the phyla that are deuterostomes are Echinoderms and chordates like us, right? So we are chordates. Echinoderms like sand dollar, starfish, and sea urchin have the same kind of development that we do. So we share a major evolutionary history with them, and that we can learn things about our own development by studying sand dollar or sea urchin. All right, so we have some examples to look at of slides today of the development of sand dollar or sea urchin. We also have a couple slides to look at of frog, and we have some pictures to look at for chick. Right, we're going to look at chick later in development. We're going to look at sea urchin or sand dollar. We're going to go from zygote to larva. Frog, we're going to look at sort of the middle of this process. And we see here that we have a frog embryo. Now, all this yellow is yolk. So it has a lot of yolk. Now, the chick egg has even more yolk. And it turns out that when we look at an egg, if we crack open a chicken egg, we see all the yolk, we see all the white. If there were a developing embryo in there, it would start out, it's a tiny little dot. It's about the size of the head of a pin, depending on how old it is, but it's sitting on top of the yolk and it's very flat and small. So it's sort of really squished this way. We try and look at early development in chick. The embryo is very distorted. It's really hard to see what it looks like. So we aren't looking at very early development. We're going to look at later development where the formation of body structures and organs is starting to be visible, right? So we're not going to look at blastula and gastrula in chick. Now, frog, once it goes past gastrula, so we're going to look at frog after gastrula. And this is a picture of a frog embryo that's going through gastrulation. So this is the archenteron. And it's starting to push into this ball. All right, this is the blastocele and it's disappearing. It's going to get filled in with more yolk and with mesoderm. All right, and then what we're going to actually look at in frog is the beginning of organ formation. So when we do look at very early development in frog, because it has so much yolk on one side of the egg, the cell division is happening at the other end of the embryo. And when it does form a blastocele, it has this half moon shape rather than an open circle like we would see. So we can see that there's differences between frog embryo and sea urchin embryo, even by the time it gets to blastula. But what we're going to do is we're going to focus on a little bit later in development, and we're going to look at the formation of this neural crest. So cells start to migrate up towards the top of the embryo, and we're going to form the nervous system by having this ridge of ectoderm rise up. So some of these cells from the ectoderm, the blue, are migrating towards the top of the embryo, and they form these neural folds. Now, if we look at this cross section through the embryo, we're going to see that it starts to have kind of this two-humped camel sort of look at the top. And then the space down here is the archenteron, or the gut. The blastocele has already disappeared. Right? So we have the gut there, and that's a fairly large hole. But notice that these two humps, or this neural fold, has risen up, and there's this dip in between them. That dip is called the neural groove. And so we say that this embryo is at the neural groove stage. We look at it, we see that that is what structure it's growing right then. We say that's the stage of the embryo. So those two lumps on either side of that groove, what ends up happening is they continue to rise up until they meet together at the top. And so we end up with sort of those two humps rise up together. And now we have another space or tube that's going down the length of the body, and that ends up becoming the hollow space that's inside our central nervous system. The ventricles of the brain and the hollow nerve cord are our spinal cord. So this is called the neural tube. Oops, and I don't know why this line just drew, I apologize. The neural tube, this guy is still the gut, just like it is in that upper picture. All right, so we have slides that show this. Now, when we start growing body organs, right, this process of development, remember we had a series of steps that we could lump together and call cleavage in development. As we have a set of steps where body organs are starting to form during development, we say we're in a phase or a period of organogenesis. So organs are being built, organogenesis. All right. So the last organism we're going to look at some developmental characteristics for is the chick. So as the chick is growing inside of the egg, it has the embryo. It's going to have radiating out from it these blood vessels. So it's going to look red when we open up the egg and we see. We have a heart that's going to be in this region. We have an eye right here. This part is the brain. And then we see these little square bricks that are going down along the back. So what we have down the middle of the back is we have a structure called the notochord, which is sort of like the spinal column, but we'll talk about it more later. And then if we're looking not from the side, but from the top, we see these little bricks that are growing down the side of the notochord. So these little bricks are called somites. As development proceeds, the embryo is going to get more. So one of the things to look at when you're looking at the chick embryo is to count the somites and see how many it has. And we think about this big muscle down the back, both sides of our back. You can imagine that those somites kind of merge together and turn into those muscles that grow along the back. They're muscle tissue. Let's see what else. Okay, all right. So when we think about what is inside of a chicken egg, it is a little space capsule that is like a seed. It's going to have everything inside of it that embryo needs to get to the point of development where it can come out of the egg and live. So the white part of the egg is going to be full of protein. Like if you're weightlifting and you want to add more muscle, you're going to want to eat a lot of egg whites. It has water in it as well. The yolk is going to be filled with all the other nutrients that you need, especially fat, for energy. Then there's this other membrane that goes all the way around the embryo, and that is going to be protecting it and keeping a nice moist habitat around it as it's developing. And that one is called the amnion. So there's a space inside this membrane. The membrane is called the amnion. It has amniotic fluid inside of it. So when we look at the way that mammals have development, we have taken this structure that's sort of inside the egg here, and we keep it inside the uterus of the female. So instead of having connection to a yolk and a white, there's a connection directly to the blood supply of the mom so that nutrients and materials can get exchanged and transferred. There still is an amnion around the embryo because inside the uterus, it still needs to have a watery environment protecting and surrounding it. But just before the baby's born, that membrane breaks and all the water that was in it comes out. So a woman knows that she's pretty close to the time when her baby's going to be born, when it seems like all this water comes out of her. So that's the amniotic fluid. All right. So because we have sand dollars and we're talking about them today and we're talking about their development and sea stars, we're also going to do the phylum Echinodermata today. I know this is a long lecture, but this part isn't very long. Echinodermata. I'm going to quickly go through their very strange body organ systems, right? And we are going to start by saying they are triploblastic. They have all three tissue layers. They have a complete gut. They have body organs for most of the functions that we're going to look at for our log-drench table. So they have some kind of arms or spines that are going to stick out. They have muscle tissues in them. And then they also have these little suction cup, translucent sort of suction cup thingies sticking out the bottom, which are called tube feet. So between the arms and the spines and the tube feet, they're going to be able to do locomotion. Their skeletal structure is going to involve these calcium carbonate plates called ossicles. And different types of echinoderms have extremely obvious sets of ossicles. Some of them are fused together into a solid ball shape, and some of them have much reduced. And when we did a tree diagram question on an exam earlier in the semester, that was one of the characteristics that we used to tell the difference between these groups. So that's part of their skeletal structure. For osmoregulation and excretion, all echinoderms are marine, and so they are just going to do their exchange of ions and water with the seawater around them. They actually have an opening to the water in the outside world called the madreporite, right? A pore is like an opening, and they can open and close this valve, and they're going to actually let seawater come into and out of their body space, and they kind of use it as their own body fluid, which is very weird. So they don't have a special body organ for osmoregulation and excretion. For gas exchange, they are going to be able to do gas exchange from their surface of their tube feet. They're also going to have kind of these little bulges sticking up all across their surface that are extensions of their water vascular system and connected also to their tube feet. So these little bulges on their surface, and the tube feet is going to be where they get their oxygen in and their CO2 out. Digestion, they're going to have a complete gut. Here we have the mouth. We can see they might have more than one stomach. They have these extensions of the stomach down into their arms. So they have these digestive glands that are filling up a lot of the space in their body that are the places where the digestion and absorption are going to occur. However many arms they have these digestive glands going into, so their whole body, you can think of it as almost one big stomach. They do have an anus, so they have a complete digestive tract. Now their bodies have their mouth down and their anus is on the top. Oh, I already did respiration. All right. We're going to identify them because they're going to be an obvious large organism. They have radial symmetry as adults. We probably are going to see arms. We're going to see spines. And there's a couple different body types that they may have, and we'll look at examples of those today. Their nervous system is quite interesting. They do have a brain-like structure. Their nervous system isn't shown on this diagram, but we can see that they have the central stomach, and then they have digestive glands radiating down into their arms. We'll see also that this water vascular system, shown in orange, radiates down each arm. Their nervous system has the same structure. So they have a nerve ring, which functions as their brain. Their brain is just ring-shaped instead of being in one blob. And they have nerve cords down each arm. They may have sense organs at the tips of the arms, such as able to tell differences between light and dark. And they can tell if things are sitting on top of them, like if there's a algae on top of their spines, they'll move it across, they'll move it off. So they do have a nervous system that reflects their overall body pattern in the structures that it has. Their circulatory system is this water vascular system that is composed of the ring canal, the lateral canals, the ampullae, and even the tube feet are part of the circulatory system. So the whole thing together is called the water vascular system. They're able to push that water into those tube feet and extend them out and do basic body movements with them, including suction onto things. The habitat for echinoderms is they're all benthic marine organisms. So we're going to find them on the sea floor. And they can be quite common in areas that some other organisms may not be able to live in. So the very, very deep sea, the Arctic and Antarctic, there's going to be a lot of echinoderms living on those places. All right, so their bodies are very strange compared to ours. They have this radial symmetry, and often it's in 5 parts. And so sometimes we call it a pentaradial symmetry. Right, so we see 5 arms on this starfish. There are also 5 groups that we're going to talk about. So we have this class called Crinoidea, which has a tremendous fossil record. So this one is an example of what a fossil would look like. Modern day crinoids, you see they have many arms and they're sticking up. So a little cartoon of what a crinoid is going to look like, feather star sea lily, is that its mouth is going to be on the upside rather than the pointing down side. And they're filter feeders. They're going to be grabbing little particles of food out of the water and then passing them all down to their mouth. So we see them in coral reefs. We see them in the deep sea. We don't have any nice preserved specimens. We do have fossil crinoid plaster casts that you can look at in lab. Our next group, the Holothuriidea, also called sea cucumbers, and their body is quite different, right? If we imagine that the body of a starfish, although it has legs extending out from the side, the middle part is kind of a ball, right? So if we turn that ball on its side and we extend it, we have the body plan of a sea cucumber. And it has tentacles, which are modified tube feet sticking out from one end. But if we looked at it from its behind, we actually see that it has 5 rows of tube feet along its body. And we see that it does have that ancestral 5-part radial symmetry. Right, so they have an elongated body, and they're on their side compared to all the other organisms, and their body's squishy because the ossicles are highly reduced. And when they're disturbed, they'll pull their tentacles in so that they do kind of look like a lumpy sea cucumber, although there aren't any green ones that I know of. So they're often brown or black, or they can be red or yellow. This one on the top right, it has its tentacles out. And it's going to have very little obvious similarity to the other echinoderms. Our second to last, sorry, our third to last group is the echinoids. Echinoids include sand dollars and starfish, and they're going to be more ball shaped. They're either going to be a round ball, typically with fairly long spines, or they're going to be more of a flat disc with little flat spines that are sticking along. They have a highly enhanced ossicles, and they are fused together into this consistent shell or test. All right, so all the ossicles are fused together, and a sea urchin without any of its spines, once it's dead and everything's fallen apart, is going to look like this, and a sand dollar is going to look like this when it's white and dead. When it's alive, we're going to see that it has spines, it has some kind of colored tissue on it. Our next group is called the Ophiuroidea, and I remember that because the middle of their body looks like an O. These are brittle stars and basket stars. They have really skinny arms, right? So a cartoon of this is going to have a distinct central disc, and then the arms are going to be real wiggly, and they're going to be radiating out from the middle here. Basket stars, like the one in the bottom, it's brittle star-like, but its arms are lots more arms, and they have these cool curly cues on them. And our last group is the most familiar one. Sea stars are starfish. They may have more than 5 arms, but a cartoon of them is going to look like it's going to have those arms, and it's going to have a really thick area in the middle, and the arms are thick compared to the Ophiuroidea. All right, so this is our different kinds of echinoids or echinoderms, and we're going to have some examples in lab today to help you practice telling the difference between them.